All right, if you haven't done so already, be sure and watch the Stated Clearly video right above my short video, which is entitled, What is Chemical Evolution? It's going to give you some really nice illustrations that I think are going to really help to cement this process in your mind. The process of chemical evolution that may have produced the very earliest and simplest of cells likely proceeded in a sequence of stages, many of which can be replicated in the lab. In the first step, small organic molecules, that is to say, carbon-based molecules, are synthesized or created abiotically, meaning they form in the absence of life, without life forms producing them. So in the video, you witnessed that little cartoon rendering of a very real lab experiment where fatty acids will begin to self-assemble from carbon monoxide and hydrogen gas in the presence of clay silicates, like we find in those subterranean chambers in the video. The fatty acids are then belched into the aquatic environments where those molecules would be selected for, at least for longevity, because they're going to float. Eventually, those fatty acids are going to become so concentrated from repeated cycles of heating and cooling and the belching of more and more small fatty acids, because of their chemical structure, their hydrophilic heads, water-loving heads, and their hydrophobic tails, they're going to stick together and form these larger macromolecules, which again, we can produce all of this in the laboratory. These macromolecules are going to form balls, and then eventually they form these massive sheets or skins, these films of fatty acid. When the films fold over on themselves, you now have what scientists call a protocell. So what's a cell membrane made from, at least today? You probably remember from 181, you've had it drilled in your brains, the phospholipid bilayer, 2 layers of fatty acids. So this protocell is essentially now just a primitive cell membrane. And with this fatty acid cell membranes, protocells now have an internal environment which is going to differ from the external environment based upon what they captured initially and what is allowed to pass through, what is internalized. So you can envision different protocells with different combinations of chemicals internalized. Nature would begin to select either for or against certain protocells depending upon the molecules they may have taken inside. And then in the fourth step, and for me, this is really the biggest leap, comes the self-assembly of the self-replicating molecules, some sort of proto-RNA. And I've got another Stated Clearly video below about the RNA world. Remember, RNA is just made from one strand, and it is going to precede DNA, which is made from 2 strands.